Pets and Livestock. My name is Bob Schwartz, and I serve as Director of Special Projects for MU Extension's Business Development Program, and I have the honor of moderating today's webinar. Thank you for joining us for an event we've been looking forward to. And let me go through just a few housekeeping details before I introduce our speakers and we begin today's program. First, my thanks to Amy Patillo for her work in putting together the Workforce webinar series, including today's webinar. This webinar will be recorded, so you'll be able to access the webinar and slides following the program. We have about 30 to 35 minutes of presentations, followed by about 20 minutes for questions. If you would, please go ahead and upload your questions to the chat room or on Facebook. Amy will take those and move them over to the Zoom chat where we'll see them there and I'll share them with the group. With that, let me introduce our two speakers. First, Dr. John Middleton holds a BS, DVM, and PhD from Washington State University. After graduating from Washington State with his DVM, Dr. Middleton worked in private practice for about 2 years before returning to Washington State University to complete a PhD in clinical residency. He is a diplomat of the American College of Veterinary Internal Medicine, Large Animal Internal Medicine Specialty, and an associate member of the European College of Bovine Health Management. Dr. Middleton has been a faculty member in the Department of Veterinary Medicine and Surgery since 2001. And is currently a tenured professor with teaching, research, service, and administrative responsibilities in the College of Veterinary Medicine, including being the assistant director of the Agricultural Experiment Station. He is the chair-elect of MU's Faculty Council. His research is primarily focused on disease epidemiology and dairy cattle, specifically focused on staphylococcal mastitis, although he collaborates across multiple species and disciplines, including establishing livestock models for translational research relevant to human diseases and disorders. And I believe I missaid mastitis, it's mastitis, so excuse me for that. Dr. Leah Cohn graduated from the University of Tennessee with both a BS in animal science and a DVM degree. She completed a rotating internship in small animal medicine and surgery, followed by a residency in small animal internal medicine at North Carolina State University. Dr. Cohn also earned a PhD degree in veterinary microbiology and immunology at North Carolina State. After completing a postdoctoral fellowship in cell biology, Dr. Cohn joined the faculty at the University of Missouri College of Veterinary Medicine. She is currently a professor of veterinary medicine and serves as the director of graduate studies and the associate department chair in veterinary medicine and surgery. Dr. Cohn has been very involved in her specialty college as well, serving a full term on the Board of Regents of the American College of Veterinary Internal Medicine, including time as the president and chairman of the board. Her research interests are varied, but infectious and respiratory disease are particular favorites. Dr. Cohn has authored more than 130 scientific manuscripts in peer-reviewed journals, has written dozens of book chapters, and is the editor-in-chief of the textbook Clinical Veterinary Advisor, Dogs and Cats. And with that, Dr. Middleton, the floor is yours.
Thank you, Bob, for that kind introduction. So our goal today is to give some background on COVID or SARS-CoV-2, and then hopefully give you some insight into how this affects people as well as animals, and then we'll take some questions. So I've got a series of slides, and then Dr. Cohn will follow after I do. So really what I'm going to give this afternoon is some background and then look at some of the implications for animal and human health. And so the topics that I'm going to cover is a backgrounding on emerging disease threats. So I'm going to go a little bit broader than COVID, but that's to introduce the topic of where these things come from. I'm then going to give a brief history of what we know about coronaviruses, and then information specifically on COVID-19, and then what are the implications for livestock producers. So broadly speaking, COVID or SARS-CoV-2 could be considered a zoonotic disease. In other words, it has its origins in animals and as then through a series of steps become a human disease. And so a zoonosis is a disease that can be transmitted to humans from animals. And perhaps of interest then is that over 60% of human infectious diseases probably at some point had their origins in an animal. And so the graph over on the right-hand side just shows that rodents and apes, as well as bats, seem to be a reservoir for many viral agents that affect both animals and people. Some of those can be carried through to animals and people through vectors such as ticks and mosquitoes and other insect vectors. But what we see on this graph is these viruses can circulate for a period of time in these wildlife hosts, but if they happen to spill over into the animal population and we get replication in the animal population, that can then spill over into the human population, and if we get perpetuation of the disease in the humans, we get something that looks very much like what we're seeing right now with COVID-19. The graph at the bottom then shows what happens if we can forecast these viruses in their original wildlife host, detect them early, put control measures in place, and you can see that we reduce that disease spike in people dramatically, as well as we eliminate this green part of the curve in animals. And so the next slide then sort of takes that concept and talks about it. And various agencies around the world, including the United States Agency for International Development, have been looking at emerging disease threats and saying, can we identify these disease threats before they become a problem for not only animal populations, but human population? And so they have a group that works on pandemic influenza, and these are the countries on the left-hand side that they work on with pandemic influenza. But probably more interesting to the point of the discussion today is looking over on the right-hand side. These are the countries in which scientists have been collecting samples from bats, rodents, and monkeys, and studying the virome. In other words, what viral pathogens can we find in these three particular potential reservoirs for animal and human disease? And you can see it's pretty widespread, the areas of the world that they've been working in. And so this was a $100 million project funded by USAID that was actually refunded for a second iteration. The picture at the top here on the right-hand side just shows some of these so-called virus hunters in various places around the world, rainforests and jungles. And what they're doing here is they're collecting bats from caves and other environments that they live in, and they're actually collecting swabs for virus detection. And you can see they're wearing quite a bit of PPE, but that's how we discover where these viruses are coming from. And in fact, as we'll see in a moment, SARS or SARS-CoV-2 or COVID-19 actually probably had its origins in a bat. So a brief history of coronaviruses. Not known at the time, but perhaps retrospectively, in 1912, some German veterinarians examined a cat, and Dr. Cohn's going to talk to you more about the cat situation, with a swollen abdomen, and it's thought that maybe that was the first case of a coronavirus in an animal. In the 1960s then, in the United Kingdom and the United States, two viruses were isolated with crown-like structures from humans with common colds. And these viruses, similar viruses, were then also isolated from sick animals. The interesting thing is with technology and electron microscopy, when they looked at these viruses under the electron microscope, they had a structure that looked like the sun's corona, which is why they became called coronaviruses in 1968. And we have various different coronaviruses in the intestine as well as the respiratory tract of animals and people. And in fact, the ones that cause the common cold, at least historically, tended not to be very aggressive, and they were thought to just cause common cold-like symptoms, and therefore were not a huge concern because we have other viruses, rhinoviruses and so forth, that cause the common cold. And then in animals, in the food animal area, in livestock, we've had enteric or intestinal coronaviruses for years that cause diarrhea. There's a little bit of evidence in cattle for respiratory coronaviruses. And so we've had these viruses of this family, so to speak, around for a very long period of time. And certainly in livestock, we do see very severe cough diarrhea associated with coronavirus. That's a different coronavirus then than the one that we're talking about today. Then in 2003, we saw an outbreak of what was termed severe acute respiratory syndrome, or SARS. And many of you probably remember seeing that in the news, and that was caused by a coronavirus. So the original SARS was SARS-CoV, because at that time we only had one SARS coronavirus. And in 2003, there was by today's COVID standards, a small outbreak in various parts of the world. And at that time, it was thought that SARS-CoV was caused by recombination of a virus that was found in bats. So you'll see in this picture on the right-hand side, bats and rodents feature in these viruses. And these coronaviruses then went through an intermediate host. The intermediate host in this case was this civet. Civet in some parts of the world, in Asia, for example, are part of the diet. And so these are consumed by people. And that then led to this virus going from a bat, metamorphosizing to some extent into civets, and then moving into the human population. So it became human adapted and then spread from human to human. And so if you look on this chart, these are just various coronaviruses that affect people and animals. And you can see that some of these have different intermediate hosts. There's a human coronavirus here that probably went through llamas. There's one that went from a rodent through a cow. But when we're talking about SARS, it actually went from the bat into the civet. This jump here is not entirely understood, and then from the civet into people, and then became a people-to-people problem. But notably, there was only about 8,000 cases worldwide of that particular disease. Then in 2012, you probably remember seeing in the news that people were getting a respiratory virus from camels, and that was termed Middle East Respiratory Syndrome, so MERS. And that one is again thought to have come from bats based on the phylogenetic analysis of the virus, probably got into camels as an intermediate host, and then spread from camels to people. It's not entirely clear how camels infected people, because people that worked with camels never actually became symptomatic in most cases with the disease, but it is thought that camels transmitted it to people that worked with the camels, and then the camels transmitted it, the people that became maybe mildly infected transmitted it to other people, and then it was perpetuated. What's interesting about that disease is there's only about 1,000 cases recorded worldwide. So again, something that could have spread into a pandemic, but kind of died itself out quite quickly, much like the original SARS virus. So then we get to today, and SARS-CoV-2, which is the sudden acute respiratory syndrome coronavirus 2, as it's been named, is the cause of coronavirus disease 19, or COVID-19, and that's because that was discovered in December in China. And in this case, again, the phylogenetic analysis would suggest it probably came from a bat. The putative intermediate host in this case is a pangolin, which if you've ever seen a pangolin, it's kind of a armadillo-looking creature, and probably associated with these pangolins been sold in a live animal market in Wuhan in China. And then the virus makes the leap to people, and then it becomes a people-to-people problem. And to date, we've got over 6 million cases of this and counting. So this virus is different from the other two coronaviruses that have caused respiratory disease in that it's highly contagious. And part of that is probably because this particular coronavirus tends to hang out in the upper airways for a longer period of time than the other two coronaviruses, which tend to descend into the lungs quicker. And we also know that this virus is shed in very high numbers in respiratory secretions. And that then facilitates its movement through droplets between people. Now, one of the things to recognize about viruses is, unlike bacteria, which many of us are familiar with, bacteria can reproduce outside the host. So you can put bacteria on your kitchen countertop with the right growth conditions and some nutrients, those bacteria will proliferate. Viruses, on the other hand, can contaminate surfaces, but viruses require host cells to replicate and perpetuate the disease. So at some point, that virus is going to die on that surface. It's not going to replicate, and therefore that surface doesn't become a source of infection anymore. But we do know that some of these surfaces can be heavily contaminated, and the virus will survive on those surfaces for extended periods of time, in some instances probably a few days. So the primary transmission then is from person to person, primarily through respiratory droplets, although there's some preliminary evidence to suggest that it may, we can find virus, at least with some of the tests, in the air for longer periods of time, suggesting it may be aerosolized as well. But the primary respiratory droplets are thought to be the main mechanism of transmission at this time. So with that in mind, how do you protect yourself? And you've probably all seen these things in the news. But part of the reason for the 6-foot distancing is your respiratory droplets, at least with normal speech, are not going to travel much further than 6 feet. Now, we do know that as you raise your voice, that can increase the spread of droplets. And there's an example of a case report, for example, in Seattle, where a choir practice, they would distance 6 feet and people still got infected. And that's because as you speak louder and project more, your droplets travel further. So distancing is important to reduce your exposure to the virus. Washing your hands is incredibly important because as we touch those contaminated surfaces, we contaminate our hands, and just human behavior is to touch your face very frequently, and that's been studied, and people can do that many, many times in an hour. So if you wash your hands before touching your face, you're going to reduce transmission of viruses that you came in contact with on surfaces, doorknobs, bathroom facilities, and so forth. Washing your hands with soap and water for 20 seconds does in fact kill the virus. So avoid touching your face, eyes, and nose. There's now some evidence actually that if you're standing close enough to somebody, respiratory droplets can actually get into the space around your eye and cause infection. And therefore it's important that we cover our mouths when we're coughing or sneezing, because as I said, as we increase the projection of droplets, we're going to move those over bigger distances. And as we sneeze or cough, we want to not cover our sneeze or our cough with our hand, because again, our hand comes in contact with lots of surfaces as well as our own face. So use the crook of your elbow or use a tissue to protect others from that sneeze. And then if you are showing clinical signs, which can include shortness of breath, a persistent dry cough, fever, chronic fatigue, things that you would associate with the flu, or if you have a temperature over about 100 degrees Fahrenheit, then those are indications that maybe you should consult medical advice and get tested for COVID. One of the things that you've seen come out in the news over the last month to 6 weeks is this concept of face coverings. And there's a lot of debate over face coverings, but what we do know from the science and also some mathematical modeling is that if you wear a face covering, it's not necessarily protecting you, because things can be inhaled around the side of the mask, but what it does do is it stops your respiratory droplets as you sneeze and cough and speak loudly moving to somebody else because they get trapped or slowed down by the mask. And so in some mathematical modeling that was done in New York City, we would have reduced the spread of the disease by anywhere from 30 to 70% by either wearing a cloth face covering or actually a surgical type face mask. Now, currently, we're not recommending using surgical masks because they're in short supply for medical professionals that need them. So in public, we're recommending cloth face coverings. And as the CDC sign says here, your cloth face covering protects them, their cloth face covering protects you, and therefore, if we all participate in this, we're going to reduce transmission. And this just illustrates then that very point. So this is some light scatter imagery that was done in Japan. And these are two people that you can't see in the picture, but what they've done is capture their respiratory droplets with this light scatter image. And this is a person not wearing a mask, and this is a person wearing a mask. If we consider that those droplets have virus in them, you're much less likely to acquire an infection from the person on the left wearing the face covering than you are from the person on the left. Sorry, I may have got my left and right confused there, but the picture on the right says what the mask does. The picture on the left says if you're not wearing a mask, you increase the risk of infecting somebody else. So what about SARS in domestic animals? And Dr. Cohn is going to spend quite a bit of time talking about companion animals, so I'm not going to touch on that. Suffice it to say that SARS-CoV-2 or COVID is a reportable disease in animals. And the World Animal Health Association, or the OIE, currently has no reported cases in livestock. So in the previous slide, there's no cases in livestock there. These are all in cats or dogs in all of these situations. Experimental infection, there's a paper in Science in April that looked at poor viral replication in various different species. Not any work that I found yet in cattle, but in pigs, ducks, and chickens, there's poor viral replication. So if you expose these species to the virus, it doesn't replicate very well. Cats and ferrets are a different story, and I'll let Dr. Cohn talk about that. So really for the livestock industry, it becomes an indirect consequence of the disease. And as you're all aware in the news, this has to do with supply chain disruption. So you might ask, well, why is the meat processing system so um, at risk for this situation? And so if we go back to the slides on how we prevent getting infected, and then we look at these pictures on the right-hand side, it becomes pretty obvious in these situations that disease transmission or mitigation of disease transmission becomes a very difficult concept. The other thing that you will note here is that these individuals working in this processing facility are wearing beard covers, but beard covers in no way, shape, or form stop droplet transmission because they're very porous. And so while this is part of the sanitary protective equipment, it's not protecting disease transmission. And you can see these people are standing shoulder to shoulder. And if we get a cough or a sneeze, that's going to transmit from one person to the next. Now, the next question you might ask is, if they're sneezing on my meat, does that put me at risk? Pretty much the virus isn't going to survive in the kind of environments that meat is transported in. So the food is not a huge risk factor. The risk factor here is the fact that we have people working in very intense situations, in this case with very little PPE, and their personal protective equipment is PPE, with very little personal protective equipment, and therefore we're getting transmission just by the sheer numbers of people crowded together. Other things that are happening with the supply chain is feed availability for some livestock has been disrupted because of problems with delivery supply chains. Certainly the meat processing situation is something that we're all familiar with. At any one point in time a few weeks ago, most of the meat processing facilities were either slowed down or shut down in the Midwest, and they're starting to come back up. But you can imagine then if you're a poultry producer or a swine producer where you're producing many, many animals on a regular basis, if you've got no place to take those for slaughter, you get a backup in those facilities, which then leads to the last statement. If you've got no outlet for broiler chickens because you can't get them slaughtered, you're going to have to euthanize a whole house full of chickens. Not sure how much of that's taking place, but that can be a consequence of these supply chain disruptions. And then on top of that, one has to recognize that because we close schools and because we close restaurants, restaurants and schools take a different cut of meat than what you buy at the grocery store. And these wholesale cuts are much larger and they're not very amenable to repackaging for retail sale. So now we've got all of these wholesale cuts that are not ready for retail sale, so we can't redirect where they go. And some of those have been distributed to food banks and so forth. But all of these changes in the way that we supply food, either through a restaurant versus a grocery store, have caused major disruptions in how things get delivered to your grocery store. That impacts how much people buy. And then on top of that, we've got all these economic issues of people being unemployed and not being able to afford meat. And so all of this is having an impact on the livestock industry as an indirect effect of the virus because of what it does to people and because of the nature of the way that people work in this environment. That's impacted everything downstream from there all the way to the livestock producer. And as I said, a few weeks ago, poultry producers were having to decide how they were going to euthanize several thousand birds in a house if they couldn't get them to market. On top of that, we've seen a shift in where people buy meat from. So custom slaughter facilities have started to pick up some of that slack. But also then we're seeing trends in how much meat people eat, and that will have lasting effects on the industry. So a number of indirect effects that are the fact that this is a human disease that has implicated how we process and move our livestock through our production system. So some take-home points from my section of the presentation. Over 60% of human infectious diseases have their origins in animals. Emerging human disease threats and future pandemics are likely to come from wildlife reservoirs, and three big ones are bats, rodents, and monkeys. And potentially that's going to happen through an intermediate host, like we saw with some of the coronavirus examples. Preemptive efforts to detect and mitigate amplification. You might ask, so why would we spend several $100 million going out and looking for these viruses? Well, as you saw in the last slide, the economic implications of these types of pandemics is huge, and not to mention its effect on the livelihood of people and potentially animals, and therefore if we can detect these things and put strategies in place to mitigate that bump in that curve that we saw in the first slide, we're going to be a much better place to deal with these situations. While zoonotic in origin, SARS-CoV-2 is now a primarily a human-to-human problem, although what Dr. Cohn's going to talk to you about is a human-to-animal transmission. At this point in time, livestock do not appear to harbor the disease. Hence, the main implications for the livestock industry are those indirect economic effects because of supply chain disruptions that we talked about in the previous slide. And with that, I will hand the talk over to Dr. Cohn.
All right, thank you very much. I appreciate it, Dr. Middleton. So I'm going to take on the animals that we cuddle with. These are the ones that we give kisses and we sleep in the bed with. And so we have a little bit different relationship with our dogs and our cats than we have with livestock. So what are the implications of SARS-CoV and pets? And before we move on to the implication for the pets, let's talk about animal viruses. And let's see. There, I think it's moving a little bit slower than my clicker is moving. So just for a second, coronaviruses in dogs and cats. And Dr. Middleton gave us a nice introduction to coronaviruses in a number of different species. We have coronaviruses that have low virulence and high virulence. There are coronaviruses that you barely notice and coronaviruses that make you really sick. And the same thing is true for dogs and cats. In dogs, there are coronaviruses that cause some diarrhea, and that's about it. In dogs, there are coronaviruses that cause kennel cough. It's one of many causes of kennel cough being the syndrome of contagious cough that's associated with dogs being exposed to other dogs, like in a boarding facility or a doggy daycare or an animal shelter. It's self-limiting. Most dogs with coronavirus that causes kennel cough recover on their own. In cats, too, we have coronaviruses. There is an enteric coronavirus that causes diarrhea, if anything. In most cases, the cats never even get sick. But there's also a coronavirus, a mutated form of the enteric coronavirus, that causes a disease that many of you will have heard of called Feline Infectious Peritonitis, or FIP for short. FIP in cats has historically been 100% fatal. If a cat developed feline infectious peritonitis, that cat died of feline infectious peritonitis. So here's another example similar to what we see in humans with SARS-CoV-2. Some people get the illness and they don't even know that they were sick, and other people get the illness and they die. So coronaviruses can range from something that causes very mild illness to something that causes the ultimate horrendous illness. Now, as we think about coronavirus, and again, I think my advancer is being a little slow here. Here we go. I may skip back because I got impatient and hit it again. I did. Okay, I just have to wait a second for the slides to advance. So let me jump back to this slide. With feline infectious peritonitis, in 2019, we saw really a miracle. Dr. Niels Peterson from the University of California, Davis has been working to develop ways to treat FIP in cats for years and years and years. And he has been investigating a number of drugs called protease inhibitors. One of these protease inhibitors was studied in experimentally infected cats and had tremendous results. And in early 2019, he published a study with 31 naturally infected cats with FIP. Now remember, 31 naturally infected cats with FIP would normally result in 31 cats that died of FIP. But using the drug that he was investigating, 26 of 31 cats were cured of FIP. That's 26 cats that should have died that were cured. Of the cats that did die, several of them died within a day or two, so there was really no chance for the drug to work. The drug that he was using was GS441524, which turns out to be extraordinarily similar to remdesivir that you've heard about on the news, GS5734, a drug that has had some efficacy in treating human beings that are in critical cares and ill from COVID-19. So the same exact drug that has been used in cats to cure FIP is now under investigation to use in humans with COVID. So right now, we're waiting to see if there's approval from the FDA to begin using this drug, very similar to remdesivir, but it may hold some great promise for treatment. So let's move from that and talk a little bit more directly about cats, dogs, people, and COVID. So when we talk about these, there are three big questions that come to mind. And those three big questions are, can my dog or cat become infected with SARS-CoV-2? The second question is, can my dog or cat infect me with SARS-CoV-2? And the third question is, how do I care for my dog or cat during a pandemic? And I'd just like to emphasize again, we have very different relationships with our dogs and cats than almost any other animal that we might encounter, whether that's a cow or a chicken that we eat, or a squirrel or a raccoon in the yard. We have really close relationships with our pets. So the first of these questions is, can my dog or cat become infected with SARS-CoV-2? And there are two different ways to look at this: experimental infection and naturally occurring infections. And we'll talk briefly about each of these. So as far as experimental infections goes, these would be situations in which researchers take the virus, they isolate it and have known quantities of virus and apply it directly to the animal that they're studying, usually by putting the virus in the nasal passages of the animals. You'll notice that many of the studies that you're going to be seeing related to SARS-CoV come from a preprint service. And what this means, when we do academic investigations, they go under a review process where peers evaluate the article and make sure that there aren't issues or problems or things that they think should have been done differently. And then it's modified before it's finally published. But in many cases, we can have preprints that show us the early edition of the article. So you need to take everything with a tiny bit of grain of salt until it's actually published, because things might change. However, this study was very well done. It's already available. And what they did is they went ahead and tried to infect ferrets, cats, and dogs, as well as other domestic animals, with the SARS-CoV-2 virus. And what they found was it was really difficult to infect dogs. They could infect a few dogs with a high viral load, but it didn't proliferate in the dogs. It wasn't an efficient transmission to the dogs. On the other hand, it was pretty simple to infect the cats. So the cats who were administered virus directly into the nasal passages did indeed have the virus replicate in their tissues and proliferate, and they truly became infected quite efficiently with SARS-CoV-2. So this was another study that was published as a letter to the editor in the New England Journal of Medicine. They took 3 cats and inoculated them with virus directly into the nose, but they did something different. They took these cats that they inoculated, and not only did they examine the cats to see whether they could be infected with SARS-CoV-2, they were infected, but they housed them right next to uninfected cats. So the cages were butting up against each other. The cats were not in the same cage, but they were directly next to each other. And they not only examined the cats that they put the virus in the nose, they examined the cats living next door. And it turns out that the cats living next door became infected as well. So this demonstrated that not only are cats efficient for infection and replication of SARS-CoV-2, but they can transmit that infection through aerosol droplets or respiratory droplets to other cats housed in close proximity. Now, there's another study that just came out literally yesterday in preprint form, and this new study looked again at dogs and cats. They tried to infect dogs and cats, and once again, they placed cats that were infected next to uninfected cats. Just like the first study you saw, it turns out it's very difficult to infect dogs efficiently with SARS-CoV-2. It's very easy to infect the cats with SARS-CoV-2. And just like the New England Journal of Medicine letter, naive cats housed next door became infected as well. One thing that was a little bit different about the study that just came out yesterday, they watched how long all of these infected cats were able to shed virus from their respiratory secretions. It turns out that infected cats can shed virus for up to 1 week after experimental inoculation. So that's experimental infections. We can quite easily experimentally infect cats with SARS-CoV-2. it's very difficult to infect dogs with SARS-CoV-2. But what do we know of naturally occurring infections? Well, this slide tells us the number of known naturally occurring infections and deaths. And we see that in humans, as of Saturday, which was a few days back, and the numbers are higher now, nearly 6 million people have been infected with SARS-CoV-2. And more than 350,000 of those people have died as a result of SARS-CoV-2. On the other hand, since this pandemic has begun, there are 17 cats with known naturally occurring infections to SARS-CoV-2, and none of these cats has died. In fact, the majority of naturally infected cats had a very mild illness or no signs of illness whatsoever. As far as dogs go, in the whole history of the pandemic, which has gone on now since December, despite 6 million people being infected, there are exactly 2 naturally infected dogs that have a confirmed infection and no deaths or even illnesses in dogs. None of the dogs with infections have died of SARS-CoV-2. So what about those dogs? Those dogs were both recognized in Hong Kong earlier in the outbreak. At the beginning of this outbreak, a number of animals were confiscated from infected human beings, and they were held in quarantine. The original report, there were 30 dogs, 17 cats, and 2 hamsters that belonged to people who had SARS-CoV-2 and were hospitalized. These dogs, cats, and hamsters were tested on a regular basis, and 2 of the dogs tested positive by reverse transcriptase PCR, in other words, looking for nucleic material. Now, it wasn't a lot of virus that was found in these 2 dogs, but they did find virus. In one dog, a 17-year-old Pomeranian, he was positive 5 different times over the course of 13 days. He was also positive when they considered viral sequencing, viral isolation, and serology to show that he mounted an immune response to the virus. This dog had SARS-CoV-2. He was not sick from SARS-CoV-2. He remained healthy during the entire quarantine. Unfortunately, he died shortly after he was returned to his owner, but he was a 17-year-old dog. The other dog that was positive was a 2-year-old German Shepherd dog. That dog tested positive by nasal swab, PCR, viral sequencing, and serology as well, but only twice and on 2 consecutive days. He tested positive the day they first tested him when he came into isolation and quarantine, and he tested positive the next day, but never again after that. That's it for naturally occurring infections in dogs. So out of 6 million human infections, exactly 2 dogs have tested positive. What about naturally occurring pet infections in the United States? Well, let's start with the dogs down at the bottom of the slide because I just talked about the dogs in Hong Kong. There was one dog in North Carolina who did have a mild illness, and he lived with a family in which every member of the family had respiratory disease that was confirmed to be due to COVID-19. This dog tested positive initially by PCR for COVID, but when subsequent testing was done to confirm the infection, it turned out to be negative. So in all likelihood, the North Carolina dog did not actually have a SARS-CoV-2 infection. On the other hand, there have been 2 cats, both from New York, one from Long Island, one from Orange County, who had respiratory illness. This illness was shown to be associated with SARS-CoV-2. Now notice I didn't say caused by. It's possible they had some other cause for their respiratory illness, but these cats did have a mild respiratory signs. They lived with family members with confirmed or suspected COVID-19, and they did test positive not only by PCR, but also confirmatory testing. So other types of studies that have been done, commercial laboratories test routinely dogs and cats for respiratory infections. Obviously, kennel cough is a very important thing in dogs. You take your dog to a boarding shelter or adopt a new dog from an animal shelter, and they may come back with a cough. So testing is done quite routinely. Same thing for cats. Cats get a lot of respiratory disease, and large commercial laboratories test thousands and thousands of samples. 2 of our large veterinary laboratories developed and validated PCR tests for SARS-CoV-2. They applied these tests to thousands of samples from dogs in the United States, Canada, Europe, and South Korea. Now, all these samples were submitted for respiratory pathogen testing, so one has to assume that the animals had respiratory signs, but we know absolutely nothing about whether the families they lived with had any humans with SARS-CoV-2. Regardless, none of the dogs or cats tested positive for the virus. So thousands of dogs and cats tested, none of them have tested positive. All right. One other very interesting study about natural infection, there were a group of 20 veterinary students in France who lived in a communal housing situation. 13 of these students developed clinical signs of COVID-19. In several of the students, it was confirmed. In other of the students, it was suspected because they all lived together. These students owned 12 dogs and 9 cats. All of the dogs and cats were tested on a routine and regular basis once their people became infected. None of the pets became positive by PCR. None of the pets developed an immunologic response denoted by antibodies, and none of the pets became ill, despite living in the house with multiple humans with SARS-CoV-2. So it appears the question of, can my pet be infected? If your pet is a cat, yes, it can. If your pet is a dog, probably highly, highly unlikely. But the next question, and maybe one that's more important, is can my dog or cat infect me with SARS-CoV-2? It is unlikely, but we cannot say it's impossible. There are 2 different ways that you might become infected by your dog or cat. One of the ways that a person might become infected is simply by contamination of the fur as a fomite. So if your dog or cat ended up getting sneezed or coughed on by a person who had COVID, they could put those respiratory droplets on your pet's fur. And then if you cuddle and kiss and hold your pet, you could pick up the droplets and become infected yourself. The same way we're being cautious when we handle door handles or computer keyboards or other frequently touched surfaces, they could be a mechanical fomite to transmit the virus. This assumes that somebody with COVID just sneezed or coughed on your cat or your dog, which is not very likely, especially if you have your pets kept indoors or on a leash. The other thing is fur is probably not very good to hold those virus particles. Different surfaces hold on to viable virus particles for different lengths of time. On some surfaces, they can stay viable for 1, 2, 3 days. On other surfaces, they're pretty much gone within a couple of hours. While there are no published studies on the fur as a fomite for SARS-CoV-2, there are studies on the fur as fomites for other respiratory infections. And it turns out that fur is a pretty bad fomite. It is not an efficient way to transmit infection. And in fact, there's never been a case demonstrated of a dog or a cat being able to act as a fomite for influenza virus transmission between human beings. So while I can't say it's impossible for fur to be a fomite, you'd have to have pretty bad luck for somebody with COVID to cough or sneeze on your pet, and then you immediately start playing with them and contact their fur. The other way that you might become infected is if your dog or a cat had a true infection and was shedding the virus in their respiratory droplets. Well, we've already said that dogs are difficult to infect, so that seems pretty unlikely. Cats can be infected and they shed virus. They can infect each other. It's obviously difficult, if not impossible, to do a study where you purposely infect a cat and expose it to a person and see if that person can become infected. So we don't have any experimental studies of this question, but I can tell you that there are no cases reported where it has been even proposed as an idea that a cat served as the transmitter of the viral particle to a human being. And certainly, as veterinarians, we have not seen an uptick in respiratory infections in cats. It seems to be that while cats can be infected when you place the virus in their nasal passages, it's not a common, naturally occurring scenario. So the likelihood that your cat will become infected, if it's an indoor cat and no one in the household has SARS-CoV, is nothing. If your cat goes outside and might come across a person who is infected, it is theoretically possible that they could become infected and theoretically possible that they might transmit the virus, but it's pretty darn unlikely. What's much more likely than your dog or cat transmitting the infection to you is you transmitting the infection to your dog or cat. So Dr. Middleton talked about zoonosis being infections that are passed from people to animals, but there's also something called an anthropognosis, where the reverse situation occurs. And in that reverse situation, we have people that pass on the virus or the infection to animals. This does seem to be a possibility, especially for cats. So it's appropriate to keep your pets socially distanced from people that have respiratory illness during this outbreak. Which brings us to, how do we care for our pets during a pandemic? How do we care for our pets during this coronavirus pandemic? And there are different things to consider, including the emotional, mental, and physical well-being of your pet and of you. This has been a strange time. Parks are empty, dog parks are closed, playgrounds are closed, people are home all day long during shutdowns, spending more time with their animals than they ever had. And not only are humans feeling the effects of this, but animals are feeling the effects as well. So animals, whenever there's a change in their routine, there can be a change in the animal's behavior. And certainly people are seeing changes in their animal's behavior. Let me try to get my slides to advance here. Whoop, I went a little too fast. My clicker is a little bit off kilter here. It's a little bit slow. Sorry about that. So this was an article published in Vox recently. Why is your pet acting like a weirdo during quarantine? Explained by animal behaviorists. There are a number of things to consider for why your pet is acting like a weirdo, but any change in routine has impacts on how our animals behave. Some of the impacts may be as simple as a lack of sleep. You would be amazed how much time dogs and cats spend sleeping while you are gone to work. And with you home all day, they don't necessarily get the appropriate rest. We're seeing animals brought in where owners are noticing changes in their behavior that may in fact be changes in the behavior, but it might also be a behavior they've never noticed before because they haven't spent all day with the pet. Or it may be that their pet has altered its routine and is not getting the rest that it typically gets. Or it may be that the pet is stressed, picking up on human stress and humans' behavior, because our pets are very much in tune to our feelings. And when they pick up on stress, they demonstrate something called displacement behaviors. And this might be as simple as yawning or grooming overly much or pawing at you. So there are all sorts of behavioral changes that we see in our dogs and cats as well. One thing to keep in mind, if you have a dog during this pandemic and you've been home with it, they get very used to spending more time with you pretty darn quickly. They get used to the idea that they get to go for several walks a day and they get to hang out below your chair while you work. When you just pick up and go off to work like a regular day for 9-10 hours when this is all over, that may lead to some real separation anxiety. So it's very important that right now you try to maintain some routine and consistency for your animals, and that you leave your dog for a little while every day. Even if leaving your dog means you take a walk around the neighborhood while your dog stays in the home. Get them used to you going out of the house. Don't have them spend 2 or 3 months being with you every minute of every day. So the final question is, how do you care for your pet during a pandemic as far as medical care or taking cautions? Well, you want to be a little bit extra cautious in this time so that you don't have to take an emergency visit to the veterinarian's office. So we don't want to have our dog or our cat end up running away from us and getting into a dog fight, or have our dog and cat get hit by a car. And you can avoid that by keeping your dogs on a leash, keeping them behind a fence, keeping your cat indoors. These sorts of things keep them safer all the time, but it's especially important during this pandemic that we decrease the risk of having to go to the veterinary office. Other things that you can do: minimize trips that you make to the store to pick up supplies. You can order online, you can stock up and make sure you have plenty of cat food and plenty of dog food so you reduce your exposure, because frankly, your dog and cat's most likely source of exposure is you bringing home the virus to the pet, not the pet bringing the virus to you. And one final word is to consider who would care for your pet if you were unable to do so. If you develop respiratory illness, because it is possible for you to transmit the virus to your cat especially, but even to your dog it may be possible, you should socially distance. You should not be the one preparing their food. You should not be the person who's giving them cuddles and attention. You should not be allowing them to sleep in the bed with you if you're ill and have to spend time recovering at home. And certainly, if you end up sick enough that you require hospitalization, you'll have to have someone else take care of your pet. So give it a little bit of consideration. Maybe talk to the people who you would ask to care for your pet if that happened, and then simply enjoy spending time together. There's never been a better time to take lots of walks and get out into the woods and spend some time with your dog or cat. And with that, Dr. Middleton and I would be happy to take questions.
Wonderful. Thank you very much, Dr. Cohn and Dr. Middleton. Those were remarkable presentations. I have to admit, I regret that I've never been a student in one of your classes after listening to you both. So thank you so much. So we do have a couple of questions in the chat, and I have a few others, but let me start with the ones in the chat. Dr. Middleton, if you want to try to tackle this one, there's a question about meat consumption and whether that is increased or decreased during the coronavirus pandemic.
So the data that we have on meat consumption obviously depends on sales. And so if you look at March, for example, sales were actually increasing, and that's likely because people were stockpiling things before the pandemic got fully into swing as we went into isolation and so forth. If we look at total meat consumption based on current predictions in May, meat consumption has decreased. But the bigger question is, why has meat consumption decreased? And is that because people's eating habits have changed, or is it more likely that meat consumption is declining because of lack of availability? So if you go to the grocery store over the last few weeks, the shelves tend to be empty of meat, and that's because of the slowdown in meat processing. So you've got an association there that you can't uncouple. And in other words, what is the interaction between meat consumption or meat sales and what people's actual habits or behaviors are? And difficult to uncouple those two things, but it's predicted that meat consumption is down about 9 pounds per capita per annum if we were to do the calculations today. But that probably has a lot to do with the fact that there's a lower availability because of all these meat processing facilities that have been closed in the Midwest.
Mm-hmm. Yeah, I think everyone's seen a bit of an uptick in the cost of meat when they go to the store. So it's interesting that it's not just a consumption issue, but gets back to supply chain and meat packing facilities, as you were talking about.
And for some people, this has been the case for a while. If you look at red meat like beef, it's been consistently more expensive than pork or chicken. So to some degree, economic factors are probably driving that too. I think I read today that meat prices are up 7%, and that's probably a supply and demand issue, but that could drive you to buy pork instead of beef. So the economic aspects of it are probably going to drive the market to some degree.
Thank you. So Dr. Cohn, there's another question in the chat that relates to inhaled virus COVID-19. And the question is, do we know how long inhaled virus from a human would live in the nasal passage of a dog, horse, cat, etc., that the infected patient has cuddled with? Have there been any studies in that area?
Yeah, actually, not from a human. It wasn't a direct infection from a human, but experimentally, the paper that I mentioned that literally came out in preprint yesterday, so it isn't included in my slides, but they looked at how long the virus was in the nasal passages of infected cats. So this is a paper where, like I say, they didn't house a cat with a person, but they put the virus in the nose of a cat to infect the cat. And then they looked for shedding not only in the respiratory droplets in the nasal passages, but also in the feces because it can be shed in the feces. The longest that it was viable in the respiratory tract of the infected cats was 7 days, and that's the longest. In several of the cats, the shedding stopped much more quickly. So in no cat did shedding last longer than 7 days. In some cats, it was as short as 2 days. And as I mentioned, even by PCR, which is less than shedding, if you have any virus there, your PCR can be positive. In the 2 dogs in quarantine in Hong Kong, one of those dogs tested positive 5 different times over 13 days, and he seemed to have virus present for at least 1 week. The other dog only had virus present on the day he went into quarantine and the following day. But those were naturally infected dogs, and so there's no telling how many days they may have had it in their respiratory tract before they went into quarantine. They went into quarantine when their owners got sick. So it appears that the virus in cats may be shed for up to 1 week, and in dogs, it probably is a very similar length of time.
Okay, thank you very much. Dr. Middleton, you had talked in your presentation about a lot of these viruses originating in bats, rodents, and monkeys. And we have lots, probably millions of species of animals on the planet. Why do we see so much virus? Why do we see so many viruses originating in typically those types of species?
That's a great question, Bob. Those seem to be the ones where most of these emerging viruses have been found. What it is about the biology of the bat that allows these things to incubate, and then why these viruses jump from the bat to an intermediate host. I don't think we completely understand that, but we know from these studies that they've done that those seem to be a common wildlife reservoir. And probably not surprising then that some of the other things that have caused major pandemics in time, be it viral or bacterial, have also emerged through things like rats and so forth. So if we think about some of the other things that cause pandemics, those particular animals have a high reproduction rate, which may play part of it. But the specifics on why they jump from different species, a question that came up when I gave a similar talk recently is why, if it came from bats, does it not go back to bats? And that's because the virus undergoes some morphologic changes in its nucleic acid over time so that it becomes adapted to the human species. But why did it adapt to the human species? Why is this coronavirus more virulent than MERS or SARS-CoV? And that's just how it integrates with the host. Probably one thing that's different about this version of coronavirus is its ability to be shed in very high numbers from the upper respiratory tract. And there's some interesting work out of Germany, actually, where they showed that elderly patients, believe it or not, actually shed more virus than younger patients, which might explain why we see some of these nursing home outbreaks, because the elderly seem to have virus in their upper respiratory tract for a longer period of time in higher numbers. And maybe that plays a role in these nursing home outbreaks that we're seeing. But the specifics of each of those steps are poorly understood. The pangolin is thought to be the intermediate host for SARS-CoV-2, but that's not necessarily definitive either.
Yeah, I appreciate that. Thank you for that answer. I have lots of other questions. Very curious about wet markets and what role those play in transmission, and also very curious about the testing for COVID that seems to be very similar between dogs, cats, and humans in terms of how we test. But I'm afraid we're just about out of time. To those still online and on Facebook, if you have other questions that you would like a response to, please feel free to email those to me, and I'm happy to pass along to Dr. Middleton and Dr. Cohn. My email is schwartzrob@missouri.edu. But I know we're almost to the top of the hour, and I'd just like to take this opportunity to thank all of our audience members for joining us today. And also would like to thank our 2 speakers again. So thank you so much, Dr. Cohn and Dr. Middleton. We appreciate your presentations and your responses to the Q&A.
Thank you. I appreciate the opportunity.
Thank you, Bob. Appreciate it.
Mhm.